Hey everyone, and welcome back to AP Biology. So now we're up to lesson 6. In our 5th lesson, we talked about the history of the Earth. We mentioned where life began, from inorganic compounds to organic compounds in the Miller-Urey experiment. We talked about liposomes and mice cells and ribozymes and it being an RNA world. So now what we're up to is classification, and we're going to begin by talking about bacteria. Bacteria can be broken down into 2 categories. We have eubacteria and we have archaebacteria. In addition, this formerly was known as the Monera kingdom. So let's take a look at our wonderful prokaryotes, our first cells that showed up 3.5 billion years ago. And remember, the Earth is about 4.6 billion years, so it's about 1 billion years of there being no life, the Earth cooling, and slowly these membranes forming and separation of the outside environment to the inside environment. Meet our first life. Okay, let's take a look at this. So before we start, you should definitely know about Linnaeus for the AP Bio exam and how he came up with these 7 taxons, right? And going on even further, we have now the domain that we've added on to his 7 and given it an 8th. So we have domain, kingdom, phylum, class, order, family, genus, species. Before, when I went to school, there was no domain. We just memorized a nice nifty way. King Philip came over for a good soup. And then we understood it. Now you just remember domain in the beginning and you should be just fine. Here's a classic picture of what we're talking about, where we have the kingdom Animalia for this leopard here, right? Panther. This is a chordate, meaning that it has a backbone, okay? It has a spinal cord. Mammalia means that it gives milk, right? It has mammary glands, and that's how it feeds its young. Carnivora is the order, so this means it's a strict meat eater. Bears, otters, dogs, they all fall into this. The family is Felidae, so they've come from like this cat family, right? Panthera is the genus, and then Panthera pardus would be its species. So now, taking a closer look, we have the 3 domains here. This is where we're going to focus on in today's lecture, okay? And then domain archaea, we're going to kind of like go into that in the end. And then the next few lectures, we're going to go completely over any cell that has a nucleus. But today, let's focus on cells without a nucleus and how diverse they actually are, because this is extremely complicated to have to classify. It's a really daunting thing. So just taking a look at it, 5 major classes of bacteria: proteobacteria, chlamydias, delta-protobacteria, gram-positive bacteria, cyanobacteria. Okay, but meanwhile, we can actually just start with their shapes and what's their structures about. So there are 3 that you should know: the round-shaped, which are coccus, the rod-shaped, which are bacillus, and the spiral-shaped, which are spirilla. And some examples here, if they're together, you call them diplo. So like this would be diplococci, staphylococci when they're in grapes, and in a straight chain, I call that streptococci, right? And then some others, bacilli, right? Some examples of our bacillus, these rod-shaped. We have Salmonella typhi, right? Mycobacterium tuberculosis, Clostridium tetanae, and even Pseudomonas. And then we also have our spirochetes, or spirillums, Triponema, and Leptospira. And just some examples, right? And so here's a nice micrograph of these 3 bacterial shapes. One of my favorite rod-shaped bacteria, Bacillus, is E. coli. Because why? They help us so much, right? We have to eat a wide variety of diet because of them. Great symbiotic relationship. So now that we have shape isn't even enough to try to classify bacteria. How about we do something cool, right? So Hans Christian Gram comes up with this thing called gram staining. And bacteria are usually classified as either gram-positive or gram-negative, depending on how they do this very unique stain. The stain comes from the bacteria's cell walls. Gram-positive bacteria have a very thick peptidoglycan cell wall and the standard plasma membrane, while gram-negative bacteria still have the standard plasma membrane, but a thin layer of peptidoglycan. And then on top, on the outer membrane, they have this carbohydrate portion called lipopolysaccharide. Okay, so there's lipids and carbohydrates mixed in. This special layer causes the gram stain to come out completely different. So it's great. We now have shapes, and then whether or not you turn pink or violet. So let's take a look. If you stain purple or violet, you're gram-positive. If you stain pink, you're gram-negative. And these are just the steps: fixating it on a slide, adding the crystal violet, the iodine treatment, decolorization, and then the saffron counter stain. And after that, you can basically detect whether they're gram-positive or gram-negative. Further, we look for unique structures because only certain bacteria can make a capsule, for example. Capsules are great because it allows attachment to the host. So, for example, strep throat has to stick to like the upper respiratory tract of your cells, and they're able to do that without binding to any kind of receptor. They're just sticking there, okay, to the epithelial tissue. Fimbriae kind of do the same thing. Even so, these form colonies, right? So not only can you like team up with other bacteria of your type with fimbriae, but that's what these red things are. These are like extensions, right, that bind to the surface of the cell, can bind to maybe even the host cells, right, forming colonies and so forth. Not every bacteria can do this fimbriae stuff, right? Whether or not you have a flagellum, that's going to play a role too. How many flagella? Are there multiflagella? Is it just one flagellum? That's going to play a role. And then also whether or not you have specialized membranes. What you're looking at actually here is what we're going to come back to when we start doing the cell structure in this AP Bio course, but mitochondria and chloroplasts used to be bacteria. At least that's what the endosymbiont theory proposed by Lynn Margulies suggests. And so I'm looking at here one of the earliest mitochondria. This is the mitochondria before it was engulfed by a eukaryotic cell. It began as a prokaryote. Evidence of this shows that mitochondria have their own DNA and they have their own ribosomes. I want you to see the chloroplast before it got swallowed up. This is also a type of bacteria that can do photosynthesis, and it looks like it has a whole bunch of thylakoid stackings. All right, now the prokaryotic chromosome is very interesting because all of them have one chromosome, right? Bacteria just get one circular chromosome. If you were to unwind it, okay, and I don't know if you can see it in this picture, but when you can unwind it, you actually see there's a lot of DNA there. It's tightly wound. Genes are turned on and off. Actually, they're going to be called operons when we get to that in genetics. Now, this is the coolest thing. If a bacteria can do this, it's like quite deadly. And so one famous bacteria was anthrax. Anthrax was used as biological warfare during the early millennia. And what was interesting was that it was dead when you received it in the mail, right? So this came in like the mail, like a white powder. When you opened the envelope, some of that white powder accidentally, you inhaled it. Once it hit your nasal passages, it was moist here. So these cells got hit with water, and the endospore, this important casing, disappears, and now the bacteria is active. And anthrax was actually quite deadly. I think if you got it, you died anywhere between 2 and 3 days. And you weren't sure you even had it, so that first night was crucial. Then you ran right to the emergency room and purged yourself, right? Okay, how do they exchange genetic material? One thing bacteria can do that's really cool is transformation. So basically, it's able to take up naked DNA from the surrounding environment, right? So one bacteria can die. Oh, whoops. I think I went over, yeah, I'm sorry. I didn't bring up the transformation here, but I think that we're going to talk about it more later when we get to genetics, but they're able to take up naked DNA. Another nifty way that they do get new DNA as well is through transduction. And this is where viruses that specifically only infect bacteria can inject some DNA that came from the previous host. Okay, I'm not talking about viral DNA. Yeah, viral DNA might be mixed in, but sometimes they get DNA from a host that they already destroyed, and they got some of that DNA from that host that wasn't fully broken up, and they give it to another bacteria. These are ways that bacteria can acquire new DNA. Another way is through bacterial conjugation. And so basically what they do is they form this awesome sex pilus. Oh, wow, and this guy even has fimbriae like we saw before. Okay, and they're rod-shaped, so they look like bacillus. And so they make this wonderful sex pilus where they can exchange tiny little rings of DNA called plasmids with one another. And so what we're seeing here is that this is the plasmid that allows the bacterial cell to create the sex pilus. So what it does is it makes a copy of this using its DNA polymerase and sends it in. And now, what was once the F- cell has now become the F+ cell. And so maybe it could give it other plasmids now that it has a sex pilus. Maybe it could give it the plasmid for some antibiotic resistance like penicillin or ampicillin or tetracycline. Okay, now here's another thing. Sometimes it incorporates that plasmid, that fertility plus plasmid, into its own circular chromosome, and then it becomes this HFR cell. Okay, awesome. Another way to organize bacteria is what's their relationship with oxygen and what's their relationship with energy and just to go, right? So we have a couple types of bacteria here. We have the photoautotroph, the chemoautotroph, the photoheterotroph, and the chemoheterotroph. What's the most important thing that I would really want to have on the AP exam is what's the energy source for these things and what's the carbon source. If I ever see photo, I know that light will be the energy source, just like I see here. Okay, whether you're autotrophic or heterotrophic, photo means light. Now, the other thing, if you're an autotroph, most probably you're going to use something inorganic like CO2 to take in food, right? Or just taking in the gas, because you're going to make your own food. So you can take something inorganic. Now, if you're chemo, I'd assume inorganic chemicals would come over here, okay? Except for the heterotroph where it's organic chemicals. Nevertheless, autotroph and heterotroph, they work with different things. If you're a heterotroph, you could definitely take inorganic compounds. And so, nice rule of thumb, heterotroph, organic compounds, photo means light, autotroph, inorganic compounds. You get through that and you'll be fine. Okay, and let's take another look. So also cooperation, cyanobacteria, heterocysts, we start to see them forming these awesome colonies with each other, okay, giving them better life, right? Almost reaching this multicellularity. Their relationship with oxygen, we have 3 types. If you're a bacteria that can take oxygen, then you're called an obligate aerobe, right? And you actually require it or you'll die. All right, now if you could tolerate oxygen, you're a facultative anaerobe. You prefer not to be around oxygen, but if it is around, you can put up with it. Okay, and then of course there's the obligate anaerobe. It doesn't want to be in this oxidizing atmosphere. If you put it near oxygen, it's poison to it, it'll kill it. Okay, and these are the 3 types. So already we're seeing so many ways. What's your nutrition? What's your oxygen? What's your shape? Are your grams stained a certain way? Can you make specialized structures like endospores and fimbriae and capsules, right? What's your relationship with nitrogen would be the next thing, okay? So we have some of these awesome rhizobacteria that live in the soil, okay? And they have nitrogen-fixing bacteria that can convert nitrogen, gas into ammonium. We have ammonifying bacteria that can convert dead stuff like detritus and waste into ammonium, nitrifying bacteria that can take that ammonium, convert it into nitrites, and then eventually nitrates, and then denitrifying bacteria that can take nitrates and then basically turn it back into nitrogen gas, completing the nitrogen cycle, right? Not all bacteria have this special relationship with nitrogen. So this is a nice, good summary of what I've just said, okay, so that it comes up a little bit easier for you. I like it because they've color-coded your bacteria. There were 4 types, and they go do 4 different reactions. Really, making one big stop ammonium and the next big stop the nitrate. Ammonium can go into the plant root, which is sitting over here, but it would rather do better with nitrates, which is what we find in soil fertilizer. Okay, and again, this is the nitrogen cycle. Another good look at what's happening here, whether you're having the relationship with nitrogen or not, okay? So here's some other stuff now. Now we're looking at archaea. These guys are so important, okay, because they're also here for a long time. They call them archaea because it's like ancient. Here's some crazy places they like to live because they're extremophiles. Maybe they live in sea vents, deep sea vents, right, near volcanic activity. Or yellow hot springs, right, like in Yellowstone National Park. Antarctica, subglacial lakes, right, the Atacama Desert, and even in Europa we've seen them. So thermophiles love heat. And one of the cool things is we found this one called Thermo Aquaticus that can withstand very high temperatures. All of its enzymes, surprisingly enough, don't denature at any high temperatures, right? The hydrogen bonds don't break. I mean, their protein is folded in the coolest way where it evolved, not to really rely too much on hydrogen bonding. Anyway, we use that in PCR reactions, and this is great for multiplying DNA many times over. Halophiles. Now, this is a picture of the Dead Sea. Halophiles are salt lovers, okay? Coming from the word halogen. Now, these guys can live in really, really salty environments. What should usually kill a bacteria or plasmolyze it doesn't do it to these guys. They have excellent ways of conserving their water and actually withstanding high salt concentrations. Okay, also there's symbiosis going on. We know that bacteria help us decompose, right, and get rid of the dead stuff in our environment. We also know they play a big ecological role in our digestion, right? They live in our large intestines, such as in this termite or this cow. Without them, we couldn't digest the cellulose in wood or the cellulose in grass. Okay. Also symbiosis, we've seen bioluminescence, like in the anglerfish and certain species of jellies, right, where there's just this glow that comes out of them. That's actually mutualistic bacteria that live inside these organisms. Okay. And finally, we just started to compare bacteria, archaebacteria, and eukarya. Just some things to point out that would be helpful is the word intron. Intron are like regions of the DNA that like don't code for the actual protein. And so introns are not really found in bacteria, but they are sometimes found in some genes of the archaea, and they're always found in the eukaryotes. Okay, another thing is that bacteria and eukaryotes have different ribosomes, okay? They definitely have a different way of organizing their chromosome. And so we're seeing a huge difference right here. All right, guys, this concludes this lecture. In our next lecture, we're going to go over the first prokaryotes, the protist kingdom. I'll see you next time. Be safe.